Okay, thank you for joining today. I'm Katherine Rittenbach. I'm a technical applications scientist with de novo Software, and today we're going to be covering cell cycle analysis in FCS Express 7. If at any time today you have a question, please feel free to enter it in the Q&A section of Zoom. If you can't find that, it should be in your dashboard. But you can alternatively enter something in the chat if you're not able to find that Q&A section. I have my colleague Greg Veltri on the line, and he'll be helping with answering questions throughout the webinar today. So we have a lot to cover today, so let's get started. Here's the agenda for today's webinar. First, we will cover an overview of cell cycle analysis. Next, we'll go into exploring the multi-cycle tool that's available in FCS Express. Afterward, we'll explore how that multi-cycle tool is detecting a number of cycles and how you as a user can also select the number of cycles that are present in your sample. Next, we'll be able to go over the 6 preset models for the multi-cycle algorithm that are built into FCS Express. I'm going to show briefly a demonstration of working with image cytometry data as well as the multi-cycle tool. With FCS Express, you can work with both flow cytometry and image cytometry data. And finally, I'll show you a template that we have available on our website for doing cell cycle analysis. So you are free to build up a layout from scratch, or you can choose to download our template and use that for your analysis. Okay. So let's get started with exploring the cell cycle. So everyone here, if you've taken a biology class before, you've probably have some familiarity with the cell cycle. But let's just go through this so that we're all on the same page. At the beginning of the cell cycle, cells are going to be starting in the G0 phase and then be transitioning into the G1 phase. Now during these phases, the cells are preparing for DNA synthesis. The next phase of the cell cycle is S phase, and during that phase, DNA is being replicated. Afterward, those cells are going to transition into the G2 phase, where they are growing and preparing for mitosis. And finally, those cells are going to enter the M phase, or mitosis. And afterward, all of those cells are going to be split into 2 daughter cells, and those daughter cells will be starting that cell cycle back in G0. Now, each phase is associated with a known quantity of DNA. In G0 and G1 phase, the cells have 2N DNA content, and the amount of DNA in each cell is not changing during that phase. During S phase, the amount of DNA is variable, and it's changing from 2N to 4N because that DNA is being replicated or doubled. During the G2 phase, the amount of DNA is double what it was before S phase. So this is considered 4N DNA content. And then in the M phase, the amount of DNA in each cell is not changing, and it's still 4N. After mitosis, though, during mitosis, those cells are splitting into 2 daughter cells. So before completing mitosis, each cell has 4N DNA content, but after mitosis, after splitting into 2 daughter cells, each daughter cell is going to be entering the G2 phase with 2N DNA content. So if we can quantify that relative amount of DNA inside a cell, then we can determine which phase of the cell cycle that cell is in. We can quantify that relative amount of DNA inside each cell by staining the cells with a dye that binds to DNA inside the cells. Some examples are propinium iodide, DAPI, and Herxt 33342, among many others. When those cells are acquired on a flow or image cytometer, the fluorescence intensity is going to correlate to the relative amount of DNA inside the cell. So if there is double the amount of DNA inside the cell, that fluorescence intensity detected by the flow cytometer or the image cytometer is going to be doubled as well. And after acquiring that data on a cytometer, it's typically viewed on a histogram. Now, this is an idealized depiction of the cell cycle. All the cells in G0 and G1 phase have exactly 2N DNA content. All the cells in G2 and M phase have exactly double that amount of DNA, which is going to be 4N DNA content. The cells in the S phase undergoing DNA replication have varying amounts of DNA between 2N and 4N. So that's a theoretical model. In reality, the histogram looks more like this. This is generally called the G1 peak, and this is generally called the G2 peak. Now we can have variability in the width of our peaks because there's a slight variation in that exact fluorescence intensity detected on each cell, even if they are in the same phase of the cell cycle. And this can be related to factors like experiment preparation and increased flow rate of those cells through the cytometer. We can also have variability in the ratio of G2 to G1. Now, in the ideal model, the ratio of G2 to G1, fluorescence intensity or DNA content, is exactly 2. But in reality, that ratio of G2 to G1 fluorescence intensity is approximately 2. It's typically between 1.95 and 2.05. With rather wide peaks, you're going to notice that the G1 and G2 peaks overlap with portion of the S phase. So with cytometry data, there's not a perfect separation between those regions, between the G1 region, S phase region, and G2 region. So it's not going to be accurate to simply draw gates along that histogram around each peak. So I'll show you kind of what that would look like if you were trying to do that. So I'll show this with just some rectangles here. So what you might think to do or be tempted to do is draw a gate around this peak here and draw another gate around this peak here. And then draw another gate, a third gate for the S phase of the region in between. However, that would not be completely accurate. We would be including some S phase cells in this gate, and we would also be including some S phase cells in this gate. So our reported percentage of cells in the G1 gate and the G2 gate would be overestimated. Now, to determine the percentage of cells in each of these three regions, G1, S, and G2, then cell cycle analysis algorithms can be used. And we'll come back to these algorithms in a bit. The histogram can get even more complex with background, aggregates, and debris in our sample. So let's add that onto our histogram. On the histogram that includes background, aggregates, and debris, we have additional overlapping populations. It makes it even more difficult to determine which cells are in each phase of the cell cycle. Sometimes 2 or 3 cell cycles are occurring within the same sample. So let's add another cycling population to the histogram. When we look at that overall distribution of the data, we can see many overlapping peaks. Especially in a case like this, we'll need to use a mathematical model to remove that background, aggregates, and debris, and to deconvolute that overlapping peaks. The multicycle AV algorithm is built into FCS Express. The advanced version of the algorithm is included at no additional charge with all active RUO FCS Express 7 licenses. This advanced algorithm allows for background, aggregate, and debris removal, and it also allows fitting of up to 3 cycles. The algorithm uses the Dean-Jett-Fox model. The Dean-Jett model was proposed in 1974 and is the most well-established model. In 1980, that model was modified by Fox to properly fit a synchronous S-phase population with a complex distribution. And the modification involved modeling the data with a second-order polynomial. So let's explore how to use the multi-cycle tool in FCS Express to do cell cycle analysis. I'm going to open up a new layout. And we'll start with a single cycle because that's what's most common. So I'm going to drag my sample into the data list. And then from there, I can also drag it onto the layout. I'm going to start with a density plot. So I'm just going to draw a cells gate around my cells of interest here, do a little bit of cleanup first before jumping into the multi-cycle algorithm. So I'll draw a polygon around my cells of interest here. And we can drag that gate out onto the white space of the layout to create an identical plot to the first one, but just showing the cells inside of that cells gate. And then we can do continue our gating strategy here by FL2A versus FL2H. So if we look at the area and height parameter or the area and width parameter, we can do some doublet discrimination. So I will draw a polygon around my singlets here. So FL2 here is just the detector that was used to detect our DNA dye. On your instrument, you may be using a different parameter to detect your DNA dye. I'll call this singlets. Okay, so we've done an initial little cleanup and done some doublet discrimination. And now we're ready to view this on our multi-cycle plot. Now, before going into your multi-cycle plot, you can choose to just simply view those cells of interest on a simple histogram along your parameter used to detect the DNA dye. So this is what the histogram would look like for all events in my sample. And if we just want to display our singlets, if we have done some initial gating, we can just display those singlets on the histogram. And this is what the distribution of the data looks like. Now, a multi-cycle plot is a histogram essentially, but it also includes the algorithms that were developed by Dean Jett and Fox. So I'm going to push this off the page and zoom in a little bit more here. So if we want to insert our multi-cycle plot, we'll go to the Insert tab, choose the Fit category, and choose Multi-Cycle DNA. And then we're free to add that plot to our layout. When we add that multi-cycle plot, we are prompted to choose a parameter to display on the x-axis. We're going to pick the parameter associated with our DNA dye, and usually the area parameter, or the area measurement. So in this case, that's going to be FL2A. I'll click OK. And this is our multi-cycle plot. We're going to see several colors here and several outlines. If we want to know what each of those colors is depicting or representing, we can add a legend to this plot, just like you can add a legend to many plots in FCS Express. The way that we can do that is either right-clicking on the plot, choosing Format this plot, and then choosing the Legend category, or we can go to the Format tab at the top while this multi-cycle plot is selected, and we can choose the legend category from this format ribbon. When we do that, our format menu is going to open on the right. This is our live format menu that will allow us to format any object that's currently selected. So we can make that legend visible, and we'll be able to see what each of those overlays and what each of these colors is referring to. So our black line here is the distribution of our data. This is the actual data in our data file, the actual outline of our data. Then if we do see any blue etching, that's going to be corresponding to detected noise or noise that's being detected by the algorithm, noise, backgrounds. And then if we see a green line, that green line is the line of best fit or the curve of best fit. That is the model fit, how the model is fitting our data. Ideally, we would want that green line to be perfectly aligned with our black line to mean a perfect fit of the model to our data. Now, that's very rarely going to happen, but the model does try to get as close as possible to our data. And I'm going to go over a little bit later some tweaks that you can make in order to have the model better fit your data. Next, we have these three red overlays, and those are all of the three regions of our diploid cycle. And then we have some blue overlays over here, and that's going to be corresponding to our first aneuploid cycle. So the diploid cycle is cycle number 1, and the aneuploid cycle is cycle number 2. So the multi-cycle algorithm, in this case, is it's auto-detecting that there are two cycles here. So it believes that there are two cycles present in this sample. Now on this histogram right now, we are showing no gate, meaning all events in FCS Express. Of course, when we're using our multi-cycle tool, we can narrow that down if we have already done some kind of a brief gating strategy beforehand. We can choose to only show our cells or our singlets, whatever we have already gated on. So in this case, I'm going to show just our singlets. And then if we know as biologists, immunologists, researchers, we know in this sample we are only expecting one cycling population, and there are not two cycles present here. We're not trying to have the algorithm fit two cycles. Then we can override this auto detection feature, and we can tell the algorithm that there's only one cycle. So we can do that in the multi-cycle tab at the top. Once this multi-cycle plot is selected, we'll go to the multi-cycle tab at the top, and we can turn off auto fit, and we can choose the number of cycles or the particular special type of cycle that we want the model to use. So in this case, I'm going to select one cycle, and then the plot will auto update. So you can see now that we only have this diploid cycle, the first cycle, and it's broken into the three phases. We have one overlay for G1, one overlay for S, and one overlay for G2. Now, just to clarify again, the G1 here is referring to all cells in both G0 and G1 phase because they all have 2N DNA content. The diploid G2 phase here, G2 peak is referring to all the events that are in G2 and M phase because they have 4N DNA content or double the DNA content of the cells in this first peak. And then this S phase is going to be all of the cells in the S phase. Now the goal is going to be to report some statistics like the percentage of cells in each of these phases. If we'd like to do that, we can right-click on the plot, choose Statistics, and choose one of these statistics tables or boxes to report. The most popular one is going to be the DNA cycle statistics. This is going to be reporting the percentage of cells in G1, G0, G1, the percentage of cells in the G2M phase, and also the percentage of cells in S phase. And this table gives us some extra information as well. It can report the detected mean of that first peak, the mean of that second peak. It can also report the CVs for those peaks. It can give us an estimate of that G2 to G1 ratio that we were talking about earlier. And like I mentioned before, in an ideal world, it's going to be 2, but in reality, it's going to be hovering around 2, most likely between 1.95 and 2.05. And then that background aggregate and debris measurement. So it's quantifying the background aggregate and debris present and detected in that sample. Now we can also format this plot in many, many other ways, just like we can format pretty much any plot in FCS Express. One thing that you might want to do in a case like this, where you do only have one cycle and you have 3 overlays here, maybe you don't want them all to be the same color. You want the difference between those phases to be a little bit more unique. We can go to the overlays category of the format menu, and we're going to be able to see all 6 of our overlays that are present here. These are the ones that we're seeing in our legend. So we have our black line, we have our noise background, we have our line of best fit, which is going to be green, and then we have our G1S and G2 overlays here. So you are free to change the fill style, you can change the transparency, you can make it visible or hidden, and then you can also change the colors down here. So for this G1 phase, G1 overlay, perhaps we want to change the color to a blue to make it more distinct. And then maybe for S phase, we want to change that to another color, maybe a green. And then we could leave this third one as red. And now we have in our legend the differences here. So maybe the differences are going to be a little bit more obvious for your presentation. All right. There are some other statistics that are available. We can right-click that plot and go to the statistics option, and we can also show our DNA experiment statistics. Now, this is going to give us some additional information, like saying that the S phase confidence is good. This is acceptable. So we're going to be, as we're maybe making some changes to our multi-cycle algorithm and choosing a different model, we're going to want to make sure that that S phase confidence is good. And then we're also getting this additional metric here, which is chi-square. Now the chi-square value is going to be a measurement of how good that fit is. How well is the model, that green line, fitting our data, which would be represented by the black line. So it's really a measurement of how close is that green line to our black line. So in general, a lower chi-square value is going to indicate a better fit of the model to our data. And then the last type of statistic that is available here is the DNA model summary statistics. And this gives us another table, and this is showing us the 6 preset models that are available. You can access those by clicking on the multi-cycle plot in the multi-cycle tab, and you have those 6 models available. And I'm going to go through those each in a little bit more detail toward the end of the webinar. In general, when we are trying to pick a good model for our data, we can pay attention to those chi-squares. The chi-square values are reported here for each of our preset models, and you are going to be wanting to have a relatively low chi-square. So these are going to be telling you whether a model is having a pretty good fit for your data. Right. Next, we can discuss debris removal in cell cycle analysis. Now, most users probably think that it makes sense to run a multi-cycle algorithm on a completely cleaned up data file and that they should remove as much debris as possible manually through gating. Now, this may be helpful to an extent, but it's important to note that the multi-cycle algorithm actually uses the debris to help with curve fitting. So let's take a look at an example. Here we have a histogram that's displaying all the events in my data file. And we can see a peak on the left over here and a peak on the right that contains aggregates. When all the events are included on our multi-cycle plot over here, our BAD or background aggregate and debris measurement is 17.98% of all events. And the calculated S phase percentage is 7.45%. Now let's see what happens when we clean up our data and remove the debris and aggregates from our multi-cycle plot. So one way we can do that is by having a marker here, and that marker was converted to a gate. We can display that no debris gate on our multi-cycle plot to only show what is inside of this marker. And then we are free to go and cut out those aggregates, and we can cut out some of the noise or background that's over here on the left. And as we do that, then we're noticing that our model is changing. What we'll see here is that our background aggregate and debris measurement has dropped to 0. So because we kind of cut out some of the background and the aggregates, now that model and the algorithm is no longer able to detect the background and the aggregates and debris in the sample. So it's just reporting a measurement of 0 here, 0%. And what happened to our S phase is that the estimated S phase percentage shot up to 12.62%. So the model is probably not fitting our data correctly, and the S phase percentage is now being overestimated. Now, if you wish to clean up your data before using that multi-cycle tool, we would recommend doing that on a two-dimensional plot with forward or side scatter parameters. Doublet discrimination can be done before using that multi-cycle tool, but again, we recommend drawing your singlets gate on a two-dimensional plot using area, height, and width measurements instead of trying to simply cut off on the left and the right side of our standard histogram along the same parameter that we're going to be using for our multi-cycle plot. Next, we'll explore samples with multiple we'll explore samples with multiple cycles or unique properties. So by default, the multi-cycle algorithm auto-detects the number of cycles present in the sample, and it performs curve fitting automatically. So this is the auto fit feature. And I showed you that before. When we click on our multi-cycle plot and we go to our multi-cycle tab, we're going to be seeing that auto fit button. By default, when you insert a multi-cycle plot, that auto fit button is going to be activated or active and enabled. Often the algorithm chooses the number of cycles correctly. However, there are cases in which you, the researcher, have a better understanding of your sample and the number of cycles to expect. And that auto-fit feature is just looking at your data mathematically. The chosen model may not be the most accurate for your data. Here we have 6 different data files, each with different properties. In the top row of plots, that AutoFit tool correctly chose the number of cycles in the data file. So for this sample and this sample, the AutoFit tool chose that there are 2 cycles, and that was correct. And for this data file, the AutoFit feature chose that there are 3 cycles present, which is also correct. On the bottom row of plots, AutoFit did not correctly apply a model. And in cases like this, you may need to specify the correct model to use. So we'll go through these one by one. On this first plot, it appears that there are 2 cycles present, but we know from preparing the data that there's actually only 1 cycle here. This peak on the left is a standard or a reference that was added to the sample. To make that adjustment, we can select the plot, go to the Multicycle tab at the top, and choose the correct model from this drop-down menu. So under cycle here, instead of 2 cycles, we can choose 1 cycle with the standard overlay peak. It's actually built into our software. Now, when we do that, autofit is disabled, and we're going to be seeing that overlay peak, the standard overlay peak, highlighted in yellow. And we have a single now we have a single diploid cycle of G1, S, and G2 phases. Let's go on to this second example. Earlier, I mentioned that the Dean-Jet-Fox algorithm had a modification to more accurately model a synchronous S phase. This is an example in which the synchronous S phase, it has a synchronous S phase where the S phase or the cycle was arrested in S phase. So by default, the automatically chosen model is not optimally fitting our data. We know that this is the G1 peak, this is the S phase peak, and this is the G2 peak. And so we want to actually pick a different model to use to best fit our data. So we'll select our multi-cycle plot, and in the drop-down menu up here, we're going to select synchronous S phase, which is, again, built into the software. And now we can see this is a much better fit for our data. You can see that that green line is much better matching or lining up with our black line. And we have our G1 phase here, S phase here, and G2 phase here. On this last plot, the algorithm has chosen to use a 2-cycle model to fit the data. But let's take a closer look at these peaks. So we can expand this and zoom in just a bit. So we can see with this blue peak that there are actually 2 peaks present. So this would be more accurately modeled with a 3-cycle model. We have, if we click the multi-cycle plot and go to the multi-cycle tab, we can change this to a 3-cycle. And now we're going to have this red diploid cycle, a blue aneuploid cycle, which is our first aneuploid cycle, and then we have our second aneuploid cycle, which is going to be in green. And I showed you before what one of the statistics tables looked like when we had only 1 cycle, but what does it look like when we have multiple cycles present, like 3? When we right-click on our multi-cycle plot, go to statistics and show DNA cycle statistics, we can see what it would look like. So we actually have, instead of just 1 row of statistics, we're going to have 3 rows. We'll have 1 per cycle. We have that diploid cycle, and then we have our first aneuploid cycle and our second aneuploid cycle. So we have all 3 of our cycles, and for each one, a percentage of cells in each phase is calculated. We have our CVs of each peak calculated and our means estimated for those peaks. Now, if you'd like to reference a description of all of the options from the drop-down menu up here, we have descriptions in our manual. So on this page of our manual, which you can access on our website or in the software itself, under plots and other objects and plots, 1D plots, multi-cycle DNA plots, we'll be able to see the DNA cycle and model summary reference. And you'll be able to read a little bit more about each of those cycles. Next, we're going to go through the 6 different models available, the preset models. So if we scroll down a little bit more, you're also going to see that we have a description of each of those 6 preset models to choose from. So I'm going to go through those one by one and show you an example of each. So in addition to choosing the number of cycles, there are further customizations that can be done to help that algorithm fit your data. So let's click on our multi-cycle plot and go to that multi-cycle tab. You're going to notice in addition to this drop-down menu and the auto fit button, we have these two other buttons, which are DNA fit and DNA background. Let's explore the DNA fit options. When we click on that button, we're going to be just opening our live formatting menu. So the DNA fit and DNA background here, these are just other additional categories in our format menu. You're going to notice at the top of each of those categories, there's a checkbox for deferring changes. This is useful if you'd like to check mark that checkbox, make a bunch of changes to this category, and then after you're done making those changes, apply at once. This can help with the model not automatically updating in real time every time you make a change. You might want to pause the changes, make your settings changes, and then apply those settings changes at once. Now, by default, this auto fit is going to be active. And when that is active, this checkbox here is going to be check marked, detect the fit type automatically, and all of these options would be grayed out. It just means that auto fit is active, so we don't really have that flexibility to make customizations at this time. So if you do want to make further customizations, perhaps you're finding that auto fit is not working very well for your data set and you do need to make more customizations to help the algorithm fit your data, then you can uncheck this checkbox and you are free to change the number of cycles. This is the same drop-down menu as we had over here. And then you can also make other changes here, like specifying where the center of the G1 peak is and the G2 peak. You can specify the CV of those peaks. You can specify to fix the G2 to G1 ratio at a particular value. And you can also specify an order of SVs. So this would be 0, 1, 2, or greater. And I'll show you an example of what that would look like in a little bit, what kind of to look out for with those different orders of S. And next we have the DNA background category where you can make some further changes relating to the background. So here you can specify whether to use that background fit, whether to take into account clumping or aggregates, whether to take into account sliced nuclei or backgrounds. And here you can specify the locations of your background regions and the dimensions essentially. So the way to read this is you would use these fields to make specifications about the left side of your cell cycle and your cells of interest. And then you would use these fields to make specifications about any noise or background on the right side of your cycles and your peaks. So generally you have some kind of a noise or background on the left side and something on the right side. You can specify here if you would like where the low bound is and where the high bound is for each of those regions on the left and the right of your peaks. And then if there is a peak within the noise, then you can specify that here and give the fluorescence intensity value for that peak. Now there is an additional category here of our formatting menu called position markers. Now this can be useful to point out where the G1 mean is, G2 mean, etc. So the center points of your peaks, as well as some other markers along these other specified regions. So what does this look like? Well, let's show an example. Let's say we want to show a line on this histogram where our center point of our G1 peak is as detected by that algorithm. So we can click G1 mean here, and then we can show the position marker on the plot. Now it might be a little bit hard to see here that because it's red, it's about the same color as this overlay. So let's change this to black to make it a little bit easier to see. So we'll see here within this red G1 peak that we can have this vertical line, and that's indicating that that's where our G1, the center point, is. And we can add the same thing for our G2 peak, which is going to be over here for our diploid cycle or our red cycle over here. We can show that position marker, and you'll see that that's now a red line on our plot. Now with auto fit enabled, you will not be able to move these markers to the left or to the right. But if auto fit is disabled, you may have the freedom to move it from side to side in order to tell the algorithm where a particular peak is visually. If you're able to see the peak and you want to tell the algorithm that my peak is at this point or my center point of my peak is at this certain point, you can disable autofit and grab that position marker to move it from side to side. And that can better help the algorithm to fit your data. Okay, there are 6 pre-programmed models that have some combination of those settings within our DNA fit options and the DNA background options. And those 6 options or presets are called our models up here. So model 1 through model 6. So as we switch from model to model, you're probably going to notice some changes in the way that the model is fitting your data. That green line, the green outline, line of best fit is going to be shifting slightly, probably, and then the shapes of your curves within might be changing from model to model. And like I showed you before, we have in our manual here on this page, we have a description of what each of those 6 models is corresponding to and some of the properties of each of those models. I'm going to explain those to you now. So on this page here, we have 6 multi-cycle plots, and each one is using a different model from our options here, model 1 through 6. The first one is called SLS0. This is the model that gets applied by default. And this means that background is taken into account and the zero order S phase is assumed. Zero order S phase is shaped like a trapezoid, so pay attention to the red as we go through all of these models. Pay attention to this red diploid S phase. So it's shaped like a trapezoid with a flat top, and it's pretty symmetrical. And this first model is used when that debris curve looks like a ski slope. The second model is called SLCLS0, and this has the same settings as that first model, but it also takes into account clumping or aggregates. This model is used when the debris curve is flat and cuts into the G1 peak. The third model is called SLG21S0, and this means that background is taken into account and a zero order S phase is assumed. And the G2 to G1 ratio is constrained to be equal between the diploid and the aneuploid cycle. Now, if there are 3 cycles, the ratio for the diploid cycle is going to be equal to the average of the ratios for the 2 aneuploid cycles. The fourth model is called SLCLG21S0. So you can see the name of each of these cycles in the title of these plots. And this has the same properties as the previous model, but it also takes into account clumping or aggregation. The fifth model takes background into account and is assuming a first order S phase. A first order S phase curve looks like a trapezoid with a slanted top. So look at that difference between the red diploid S phase here and here. We have that slanted top of that trapezoid. And a second, we don't have an example of it here, but for your reference, a second order S phase curve is going to be higher on the sides and lower in the middle. And that might come into play if you are making a change here in this DNA fit category when you're specifying a particular order of S. The sixth model has the same properties as the previous model, but the CVs of all the peaks are constrained. And this model can be used when the G2 peak is buried in the S phase. In order to determine that most appropriate method to use or the most appropriate model to use, we can right-click on the plot, choose Statistics, and DNA Model Summary Statistics. I'd shown you this before. It looks like this at the bottom. We have one row per model, and those correspond to these 6 models for the same data file. And so what we're going to do is look at these chi-squares reported here. And in general, chi-square being lower is going to indicate a better fit of the model to our data. So we can use that as a guidance when we're trying to see which of the 6 models might be most appropriate. All right. So next I'd like to show you a quick example of doing cell cycle analysis on image cytometry data. So if you are unfamiliar with image cytometry, this is one example of image stream data. So with FCS Express, you can load in both flow cytometry and image cytometry data. This capability of loading in image cytometry data is included in all active RUO FCS Express 7 licenses at this time. So image stream data is one type of data that you could load in where each individual cell has a set of images. We have some bright field images, we have some fluorescence images, and we also have some composite images that were created on the instrument, which is essentially an overlay of this channel and this channel. Now, in addition to image stream data, FCS Express can accept many different types of image cytometry data. Now, another example is if you have microscopy data, you can actually segment that microscopy data using an ImageJ tool or cell profiler tool, and then you can load those segmented images into FCS Express to do cytometry analysis just like you would with regular traditional flow cytometry data. You can view those individual events on density plots, dot plots, histograms. And in this case, as long as, or in with image cytometry data, as long as you have some kind of a parameter that is reflecting your intensity of DNA dye, then you're able to also use this multicycle tool. So in this case, we had stained the sample with DRAC5 DNA dye, and so this is corresponding to channel 11. We have a parameter that's available with this image stream file that we have an intensity of channel 11, and it does look like our expected cell cycle curve. We have our G0, G1 peak, we have our G2M peak, and then we have some S phase in the middle. And so we're able to use the multicycle tool even though this isn't flow cytometry data. And we're also then able to report those percentages of the percentage of events in the G0, G1 peak, G2M peak, and S phase. Okay, lastly, I'd like to show you our DNA cell cycle analysis template that's available on our website. So our website is denovosoftware.com. And once you get to our homepage, you'll be able to access the template in this way. Go to the Resources button, Application Examples, Flow Cytometry Application Examples. This first option is for apoptosis and cell death. We have that kind of a template available. But our second template that's available here is for cell cycle analysis. And you'll also notice we have a ton of other templates that our team has created. So feel free to check those out as well. So in this cell cycle analysis entry, we'll be able to click this button to download the DNA cell cycle results template. And this can open in FCS Express. So you can use this template if you have an active FCS Express license or if you are currently using the free trial. So let's take a look at this template and how to use it. I'm going to open it up on my end. All right, so this is what you're going to see when you open that template. You're going to see our startup script. This is a window that guides us through the data loading process. So it's saying, please select a file. I'll click the ellipsis here to select my file, and I'm going to select my FCS file and click Next. Here we have to select our forward scatter and side scatter parameters. So we'll select forward scatter area and side scatter area. Click Next. And then we'll select two parameters for identifying singlets. This is for doublet discrimination. So in this case, I'm going to choose forward scatter area and forward scatter height. Click Next. And then finally, we'll select our DNA stain marker. So this is the parameter that's used to detect your DNA dye. And you should know what that is when you're working with your data. In this case, we had added propydium iodide, or PI, to our sample to stain the DNA. So I'm going to choose that parameter. And after that, our data just automatically loads onto the plots. If you don't see the gate here, then you can follow the steps that are over here, and the little note over here. That's okay. It just means that we had some different parameters. Maybe here we have parameters 1 and 3 present on the axes, but then when you load in your data, you are working with parameters 5 and 8. So the gate might not be present automatically. What we would do to overcome that is go to the gating tab, choose polygon gate, and then draw your gate wherever you want around your cells. And instead of creating a new gate with a new name, we would replace an existing gate, which is the non-debris gate, and click OK. And now the whole downstream analysis is unaffected. We've just included that gate on our plot with the desired position and associated with the parameters of interest. So we're going to want that gate to be pretty tight around our cells of interest. And then we can draw our singlets gate or change the shape of our singlets gate to avoid aggregates. With DNA cell cycle analysis, aggregates or two cells that are stuck together could appear like a single cell with 4N DNA content or double the amount of DNA. And so it is important to remove aggregates from your sample so that they are not falsely uh, ending up in your G2 and M phase. Then after that's done, we are automatically displaying all of the cells in our singlets gate on our multi-cycle plot here. And the autofit is enabled, so autofit is going to try to fit our data and apply the best model. We have a legend here depicting what each of these overlays is. And then we also have a pie chart here that's going to show us the relative percentage of cells in each of our phases, G1, G0, G1, S phase, and G2M. And so here we can see we have a relatively small percentage of cells in our G2 phase and about equal percentage in G1 and S phase. And then, of course, we have our corresponding statistics reported down here in this summary table. And then if we wanted to go through the process of choosing an optimal model for our multi-cycle tool, we can take a look at those chi-squares here and perhaps pick a better model that will fit our data better. We already have some batch actions set up here. These batch actions or this batch action window can be accessed by the Batch and Export tab here and Actions and Reports button. We have some exports to PowerPoint, an Excel document, a Prism export, so direct export to GraphPad Prism, and an export to PDF. All of these are already set up. You can check mark and uncheck whichever ones you actually want to use and export. You might need to double click in order to tell FCS Express where you want those documents to be saved. And of course, you can make any further changes to those as you'd like. And then when you're ready to make your export, you can go to the Batch and Export tab and choose Run. So this is the end of the webinar. Thank you very much for attending today. Overall, we have covered that background, aggregates and debris, and multiple cycles, when they are present in your sample, it's necessary to use a sophisticated algorithm to deconvolute those individual peaks for G1, G2, and S phase, and to be able to calculate those percentages of cells in each phase. So that multi-cycle tool is built right into FCS Express and is included automatically for anyone using an FCS Express 7 RUO license. And the tool can be used on both flow and image cytometry data. After creating your multi-cycle plots, you can continue using the hundreds of other tools in FCS Express for preparing your reports and for finalizing your analysis. If you haven't used FCS Express before, welcome. If you'd like to try it out for free for 30 days, you can visit our website, denovosoftware.com, and on the upper right corner, we have a try for free button. So you'll be able to download that. No credit card is required. And as you are learning to use FCS Express or trying out this multi-cycle tool in FCS Express, feel free to contact us at support@denovosoftware.com. And we have a team of experts that are ready to answer your questions that you have. So thanks again, everyone, for joining. This has been recorded and it's going to be available on our YouTube channel in about a week or so. And then you'll also be sent the recording. If you've attended today live and registered for the webinar, you'll get that recording within a week or so as well. So I'm going to stay on the line for a little bit. I'm going to check, make sure the questions have been answered here. And if you did have any remaining questions, please enter that in the question and answer field or in the chat. Thanks.